All right. Good morning, everyone. It's a pleasure to be here today. As you may know, there are 100 trillion, over 100 trillion actually, microbes in the gut that are really redefining how we think about our bodies. And so more and more, we're learning that they're really important for digesting our food, educating our immune systems, regulating our metabolism, and even interacting with the nervous system. So my lab is really dedicated to studying how gut microbes that are resident to the body influence brain and behavior. And this is a really great chance for me because it draws on my academic experiences in microbiology and immunology here at UCLA, and also in neuroscience at Caltech. We've been fortunate that our discoveries in this space have received recognition from the academic, federal, and commercial spaces, and that we've had the opportunity to translate some of our findings to try to reach people more directly. So more recently, I've been really struck by this finding that 70 million people worldwide have epilepsy, but 1 in 3 people don't respond to any anti-epileptic drugs. So there's a ketogenic diet, which is an extremely high-fat, low-carb diet that's been integrated into the healthcare system for actually treating pharmacoresistant epilepsy, primarily in kids. It's a rare case where a diet is actually used in the hospital as a treatment. And so the efficacy is quite good, so it can reduce seizures very well, but it's difficult to clinically implement and very difficult for patients to tolerate and to maintain. And so also, one really important point is exactly how the diet works when other drugs fail remains unclear. And so we wondered whether the gut microbiome could play a role. And to do this, we raised different models of mouse models of epilepsy and used technologies to control the microbiome. We basically found that the microbiome is both necessary and sufficient for protecting against seizures in response to the diet. We then also used a metagenomic and metabolomic approach combined with the simple machine learning algorithm to identify specific bacterial species and bacterial molecules that predict seizure protection with over 94% accuracy. So with all of this metadata together, we were then able to develop a live biotherapeutic or pharmacological approach for reducing seizures, even in animals that are fed a normal diet. This was all published recently last year, and it was highlighted in the New York Times earlier this year. And so altogether, our lab is really interested in dissecting mechanistic interactions for signaling across the gut-brain axis to drive therapeutic innovation. And so I welcome meeting all of you here today and welcome new commercial partnerships. So the research that I told you about was recently used to serve as the foundation of the startup Bloom Science. It's a great SAB and leadership. We also welcome research collaborations for additional projects. So I have 3 lab members here today who will be presenting posters, LE 2728 and IL 9, and they cover a broad range of topics we're interested in the lab, including how microbiome interacts with drugs for neurological disease. So we have one poster on microbial interactions with antidepressants. We're also really interested in how the microbiome influences neurodevelopment. And finally, how microbiomes communicate with sensory neurons. And so these have applications to a wide range of neurological disorders. So with that, I'm looking forward to the rest of the day. Thank you very much for your attention.